This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 356, recorded on September 25th, 2015. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
Are you the real Dickson?
I attempt that,
or
yes.
the fake Dickson?
No. I'm not a faux Despommier. I'm a true Despommier. So
because there are people on Twitter masquerading as you.
They are. Why would they want to do that?
I'm glad you asked it. I mean, we could see imitating, Barack Obama. We could see them imitating Donald Trump.
Yeah.
But not you.
No.
Dickson, it is a pretty nice day, isn't it?
Not bad. It's mostly overcast, but it's nice and warm. Not too warm. It's not too breezy. It's not too...
23 Celsius, 50% humidity.
Yeah.
A little breezy and partly cloudy.
But we need some rain.
Yeah, we haven't had rain in a while, and it's not coming till Monday, apparently.
All of the leaves are brown. Also
joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
How you doing?
Fine. It's 79 degrees Fahrenheit, 25 Celsius. We've had an average of high temperatures in September 5 degrees above normal.
How about
It's
that?
been the most beautiful September.
You're warmer than we are.
Yeah.
That's cool.
And you're blessed this week because Steve Goff is in your town.
He is indeed.
Have you seen him?
I have not.
All right. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How's it going?
Similarly gorgeous here. There's there is some broken overcast, but it's really, really high up. So it's like around 25,000 feet. Just a few clouds scattered underneath that. So
from
very,
north central Florida, Rich Condit.
Hey there, everybody. How you doing?
How's it going? You're still in Gainesville,
good.
Huh?
I am still in Gainesville. I'm in Gainesville, for a while.
Good.
There will be a little trip that you won't notice because we aren't recording that day. But I'll be in Gainesville for a while, till around November sometime, I think.
All right.
So we got 84. That's 29 Celsius, and mostly cloudy, I guess. I hate to say it, but that's what's going on. And actually, the humidity is starting to drop around here. So we're getting into fall, which is good.
Yesterday on Twitter, Alan Dove pointed out that 7 years ago yesterday, TWiV started. First episode.
Yep.
Dickson, do you remember the first episode?
I do. We had it in my office.
Yeah, so we're
when I had an office.
7 years.
Yeah.
Today we have a guest joining us. If we don't have enough people on this episode, we have a special guest. She is from Ohio State University. She is a PhD candidate in the laboratory of Linda Safe. Stephanie Neal, welcome to TWiV.
Hi, everyone. Thank you for having me. I'm excited.
Hello.
Hi. So you were where in Ohio are you exactly?
So exactly at this moment, I'm in Worcester, Ohio, which looks like Wooster if you look at the spelling, but is actually pronounced, we say, Worcester. Oh. And located in Worcester, we're about, I would say, northeastern part of the state, but a little more south. So we are located on the agricultural research campus that Ohio State has. We're not a part of the main campus in Columbus, which is the capital. We are actually about an hour and a half northeast of there, and we do research that spans the agricultural science spectrum. We also do work with human diseases and livestock diseases, and so that Worcester is where I am.
We have a Worcester in Massachusetts, but it's spelled Worcester.
I thought you pronounce it Woo or Worcester?
Worcester. Actually, it's pronounced Worcester.
So to give you what I think is the background, because I used to live in Northeast Ohio, and my grandmother lived in Worcester for a while.
Oh, wow.
That when Worcester was founded, they wanted to have the pronunciation roughly like the British do it. So they left out all those extra letters that the Brits don't say, and it's just Worcester. Just like
a very direct
Worcester.
Midwestern approach.
Like Worcestershire sauce.
Right.
So you lived in Northeast Ohio then, you said?
Yeah, I grew up in Parma.
Oh, wow. Well, I grew up in Canton, so.
Stop it, you two.
I know.
Hey, wait a minute. Stephanie, are you in the vet school then?
Well, so it's interesting how it's set up. So while I'm physically located on the research station campus, my department is the veterinary comparative medicine department of the vet school. So yeah, I'm a part of
that's a very famous veterinary school.
There are a lot of great people. Yes.
So people may be wondering why Stephanie is here. Because you don't have to, but it's a reasonable wonder.
She's a pleasant addition to the staff.
And she's got a great voice, good podcasting
she does.
voice.
You're very, very.
Well, it's good to have a better reason, maybe more scientific one too. I'm sure.
Well, I'm not done. Now, I think that I met you because of your podcast, right? Agricultural Science Today.
Yes.
Which you do with. What's his name? Tim, right?
Tim's Weber, yes.
And Tim has been listening to our podcast for a long time, too. And he's a farm in Minnesota, is that right?
Yes, he does. He has a dairy farm in Minnesota just right outside, I think it's called Elkton. It's right outside of the Twin Cities.
Right. So I knew you from that, and you guys are both listeners. You're on episode 13. It's agscitoday.com, so you should check that out. And Mike, microbiologist and a farmer doing a podcast. Very interesting. Although you're on a hiatus since November of 2014.
I know. It's embarrassing, really. But what happened was if I could offer an explanation, Tim, he is expanding his farm operation and also building a new house. I then was preparing for candidacy, and we kind of stored away, but we're going to get back into it.
So you and I had
wait till the ground is fertile to restart it.
Oh, yeah.
Come on, Kathy. Cue the groan.
I'm waiting for Kathy.
It has to be natural. It has to be natural. So Stephanie and I had a Twitter conversation last week, and we started talking about her work, and she said, I have to go now. I have 6 lactating sows. I thought she said cows, and I said, oh, that's got it. You have to come on TWiV. We got to use that as a title. What 6 lactating sows.
That's right.
But then there are 6 of us on this episode. And that
my goodness.
I'm not lactating.
Not all of us could lactate, but
So what do you do, Stephanie, in the lab?
Yeah, so what I do is, in Dr. Lindestead's lab, we work with a lot of different viruses. We work with coronaviruses, reoviruses, caliciviruses, mostly enteric, and but specifically I'm interested in mucosal immunology in regards to viral infection. And there's this virus called porcine epidemic diarrhea virus that swept through the United States in 2013 and caused 8 million pigs to die, quite a number, and did cause a dip in the pork industry, and we're actually on the rebound. But what I'm interested in is how do the immune cells of the gut recognize a virus, migrate through the blood, and end up in the mammary gland to then give protection to babies, or the piglets in my case. So that's my main focus. So I do a lot of immunological work, some basic virology, plaque assays. Vincent, you'll be happy about that.
Plaque assays?
Plaque assays as well. So we're trying to correlate immune markers of protection to this virus, and then, of course, maybe more mechanistically, how are these cells getting to the mammary gland, what's happening to them, so forth.
Is there a vaccine in development stage?
Yes, there is one vaccine. I think Harris Pharmaceuticals, but it's right now, I think currently out there in the market, and there's some others under development. So what farmers really have done to get a grip on this virus is they would take baby piglets that have died, they would harvest, they would take their intestinal content, chop up the intestines, and basically do a crude vaccination and just feed back that intestinal material to the mother.
Huh.
And then
soon to be marketed as Scrapple-Vax.
Yeah, we could try to market that.
Or one good pig deserves another.
Right. So that's how the farmers so far have been able to get a hold of it, and it's really kind of demonstrates the vaccinology in a very applicable way.
So where do the 6 sows come from?
So right now I have a trial running where I have 6 sows. They're given different doses of this virus, and then we allow them to naturally farrow. And I'm there when they farrow. I'm collecting the first milk, colostrum, which mentioned on the previous podcast. Yep. I'm collecting milk, and I gotta say, after growing up milking cows, milking sows is not as fun. It's a little bit more difficult.
We got so many show titles here.
Oh my gosh.
What's this farrowing business?
Farrowing, yes. So farrowing is really just a term for the pigs will give birth. Farrowing is the natural farrowing pigs are just going to have their babies naturally.
Got it. So you have pregnant pigs and they're lactating, and you're looking for antibodies in the milk. One of the things you're doing.
So there's an interesting connection here, if you don't mind me interjecting.
Yeah, I do.
I think you would, but I'll interject anyway because it relates very much to what Stephanie was saying. Pigs also get infected with a virus that's termed hog cholera. Have you heard of that?
I have, but I'll let you keep going.
Okay, so hog cholera.
You're going to let him put his foot in his mouth?
That's foot and mouth disease.
Do you know what kind of virus hog cholera virus is?
You're going to tell us, aren't you?
You should know if you're going to talk about it now.
Well,
I bet Rich knows.
No,
it's got to be a pox virus.
I'm still busy looking at porcine epidemic diarrhea virus, which is a coronavirus.
Oh, okay.
Yes,
and which, by the way, has
that.
one of the most disgustingly specific names I've ever heard for a virus.
It is, yes, it is nothing but descriptive. That is
which
sure.
I suppose would be great for discussing it, or getting grants. All you have to do is name the virus, and people immediately understand the problem.
Yes, epidemic diarrhea. Give me money.
In pigs, that's just wow.
Yeah.
With a high death rate. I mean, 8 million pigs. That's by the way, how many pigs
Dickson,
do we raise a year in this country?
Oh gosh, a lot more than 8 million.
That's true. But 8 million sounds like a big economic loss, doesn't it?
It's quite a bit. I think we're at 116 million.
Right.
I think.
So in the old days, when hog cholera was...
It's not called that anymore, by the way.
Okay, what's it
called? I can give you that name
if you... Classical swine fever virus, which is a flavivirus.
Okay.
In the pestivirus family.
Flavivirus.
Yeah.
All right. So in the old days, they used to feed pig scraps from the slaughterhouses back to the pigs as a source of protein.
Trichinella.
But wait, I'm going to get to that because that did foster the infection of trichinella, but it also caused outbreaks of hog cholera. So they learned that if they could cook the waste from a slaughterhouse before they fed it to the pigs, they could prevent hog cholera. And you'll never guess what happened. They eliminated trichinella also. So that's how trichinellosis disappeared from the United States, basically, in commercial pigs, because of the control of hog cholera, not because of the control of trichinella.
Very good.
Cool.
But trichinella-infected pigs were the reason why the United States was prevented from exporting pigs to Europe, because Germany claimed that we didn't do any inspections, and therefore they were unsure as to whether our pigs were trichinella-free or not, and so they embargoed our pig trade. And as a result, I think that cost the United States a tremendous amount of money in the long run.
Which I'm sure was also a great way to protect hog farmers in Germany.
Gee, Alan, it's interesting you made that connection.
Yeah.
That is a very, very common reason, a real reason.
Yeah, so where does the term pork barrel come from?
So, Stephanie, you're from Ohio, right?
I am, yeah. I grew up in Canton, Ohio, which, if anybody's a football fan, that's where the Pro Football Hall of Fame is located.
That's right.
I've been there.
Yes. So I grew up in Canton, Ohio, and ended up at Ohio State. For my undergrad, I studied animal sciences, and I ended up in the lab of a dairy scientist. Well, he's a ruminant biologist, and we were looking at chemotaxis of rumen protozoa, and we were also looking at calf behavior, so doing some welfare studies, and that kind of got my feet wet. I was thinking veterinary school, really vet school was the only exposure I had to what I could do with what I liked when I was younger. But that lab experience just got me hooked. And I went to Virginia Tech for my master's.
That's a great place.
It's an excellent place. And it was Dr. X.J. Ming who taught the virology class there and got me really introduced to virology. I had an excellent master's mentor, Isis Konevsky Malarkey. She was an immunologist, mucosal immunologist, but it was Dr. Ming who got me excited about virology, and then I ended up here with Dr. Safe at Ohio State. So it was a good progression into what I'm doing now.
So next year, ASV is at Virginia Tech. Are you going?
Oh, of course. I'm excited. Lots of good friends there.
We had XJ on TWiV a while ago.
Yes.
Did you hear that
great
episode?
Yes, he's great.
All right. We have a little follow-up today. I wanted to point out my pick last week was a comic book about viruses by Susan Nassif, and she emailed me this week to tell me she is a finalist in the In Vitro Gen Science Hero Awards.
Wow.
She said, please have your community vote for me. So we will give you a link. It's a little funny page. You have to search for her, and then when you vote, you actually have to vote for all the other categories at the same time. So just make sure the button next to her is ticked. It doesn't matter what the other ones. I don't know who the other people are anyway. You can vote for anybody. So vote for her. See if we can give her a big TWiV bump. Also got an email from Neva, and this is probably because Rich was talking about her. Are you there, Rich Condit?
I'm here.
Can you read Neva's email, please?
Sure. Neva writes, howdy, super professors. That's us. I was late listening to The Cat in the Heart. I have been living in interesting times here lately, and was very warmed and delighted by the kind words of Rich about our lunch meeting. We had a wonderful time and great conversation about a variety of subjects with a good amount of laughter. Certainly a testament to Rich and Ibby's generous friendliness. It was much fun, and I certainly hope that we can all meet again, Rich, when you return to Austin. And Kathy, it was delightful of you to remember Buda and share the Buda generator you found in the wilds of Alaska. Thanks. That never made it to TWiV, the Buda generator. That was in the background email exchange between
right,
Neva
and it had nothing to do
So,
with Buda, Texas. It was Buda, Illinois. But
how many Budas can there be, right?
2 at least.
The Twix casts are so fascinating. I've always been interested and amazed by biological sciences. It's true what Dr. Racaniello says. You just couldn't make this stuff up. It was fun to be reminded of the From Buda to Stump Grinding episode, which I thought was hilarious. Your collegial travels down rabbit holes are quite fun. I really appreciate, too, when you each take time to broaden discussions for us non-scientists. You're all doing great work. Science education in general seems to be especially needed in these interesting times. I am puzzled and saddened by the various forms of science denial in our society, especially the anti-vaccine proponents who can cause actual damage to children and populations. The logistics of vaccine programs are challenging enough without people freaking out and rowing backwards. In my opinion, the current crop of presidential debaters display a surpassingly willful ignorance and dismissal of facts and history, but that's politics. I focus on listening to and reading about science and technology as a positive respite from the news. Keep up the wonderful podcast, Super Profs. You are very much appreciated, Neva from Buda, Texas. Thank you, Neva.
Thank you.
Yes, indeed. We had a great time. Nice letter.
I guess we should be abbreviated, huh?
We probably should be.
Speaking of the presidential debaters, we have a letter from Eric Delwart, who's a virologist out in San Francisco, who Rich and I interviewed a long time ago on TWiV. I would call him a virus discovery guy. Dear TWiVomics, did you catch that kerfuffle at the Republican debate about vaccination? Do these candidate expect parents will bring their kids in every few weeks to space out their vaccine shots? Some vaccines are more essential than others. It was probably a reference to the highly effective papillomavirus vaccines driving young kids crazy with lust. So sad seeing the most efficacious and cost-effective medical advances of the last 100 years being constantly dissed. Do they really believe that there is a conspiracy driven by the entire medical profession, not a peep from the other candidates?
I found that whole thing really weird. It's as if in order to look conservative, you have to be anti-vax, or at least, and I don't quite understand that. First of all, it doesn't really describe. It doesn't really. It's not a proper alignment because a significant fraction of the anti-vax guys are the opposite of conservative.
Yes.
But the only explanation I can come up with is that it lines up with the idea that the government shouldn't be able to tell you what to do.
Yes.
Okay. And so, it speaks to the conservative sort of individuality thing. That's the only explanation I can find.
Well, and there's also the anti-vaccine movement, I think, picked up some conservative supporters with the introduction of the HPV vaccine.
Yes,
which,
right.
Since it's a positive thing for women's health, the Republican Party is generally opposed.
Right.
So that's part of that general trend, too.
That's why I've adopted the term for when they all get on the stage together and they start dickering with each other, it's nothing more than a mass debate.
Yes.
Got it. There it was, Stephanie. You hear her moan?
I heard it. I'm so excited.
All right. Great.
I'm capturing that moment in my head.
Dickson, where did you get that? I don't believe you made that up.
Yes, I did.
You did?
I did.
Wow, it's impressive. All right, but this is awful. That if you can believe government should lay off, but not in medical issues. That's just out of it. I'm sorry, and anyone who votes for any of these people who feel that way are really should not be. You're putting people's health at risk.
Yeah,
it's ridiculous.
What they all have in common is that they don't believe that there's a cause and effect to anything. That's the bottom line. So they can't argue logically.
Well,
They're
it's
not rational.
It's part of a trend in the past decade or so of politicians of a certain ilk becoming rabidly anti-intellectual and anti-science as a part of that.
Exactly right.
And it becomes this, my ignorance is as good as your facts.
I wish they would then put their mouth where their money is and then get out of all that technology that they're depending upon to run.
Right.
Yeah, right.
Yes.
Just stand up on a soapbox out in Central Park and people will gather around you.
Alan, can you take the next one?
Sure. Bill writes, Vincent, Dickson et al. Here I am in sunny Toronto at 18 Celsius, 65 Fahrenheit, listening to the description of the proliferation of bacterial colonies and the associated bacteriophages in the baby's gut, and how the composition matured and developed to decrease the bacteriophage population to a lower steady state at 2 years, even as the bacterial mass grew. This seems to me to be the manifestation of the CRISPR immune process in the bacteria, who eventually increase their internal stored phage-destroying patterns to that steady state. As to why this does not progress to extinction of the phages, one must assume the phages also do their own pattern fiddling to grab more food, and we might also import new phages to kick butt for a while. Then I think about diseases such as cholera. Are the cholera bacteria carrying with them phages of their own that clear the decks of other bacteria by like camp follower phages, as it were, to allow the cholera bacteria unlimited Lebensraum? Do cholera bacteria carry phages as intracellular warriors that are shed as intercellular shock troops? Does cholera have another cellular immune system analogous to CRISPR that blocks the phages in our gut from killing the cholera? Or does cholera just have its own CRISPR? How much variation occurs in assorted CRISPRs? Why is cholera immune to our phages if it is? As to links, did you know Edgar Allan Poe had a cat? And he links to a parody poem, The Conclusion of the Raven.
Right. Did he know, for instance, that cholera bacteria don't really replicate much in the human host? It doesn't. So that's the reason why you can give oral rehydration therapy over a 7-day period to allow the gut tract to repair itself, and they've completely cured themselves.
Dickson,
So
I'm afraid that you're going to get in trouble.
the biology of cholera is little understood, but if you understand it, it's...
When's the last time you read a cholera paper?
not too long ago. Rita Caldwell is a...
Rita Caldwell, right,
Writes-
40 years ago.
No. More recent than that.
I'm just worried that someone's going to say, Dickson, they do replicate in the host.
They don't.
Not at
Well,
all.
we'll find out.
And there are
We'll
no
find
carrier
out.
states of cholera in any person either.
That's absolutely untrue. How
It
do you
is.
think people delivered cholera to Haiti? They were workers who were carriers for cholera who then had no symptoms, and they shed the bacteria there, and that's been proven by sequence studies.
Well,
Okay.
you can fly to
I'm not
Haiti
sure.
from Africa in a day.
No, these individuals were shown to have the bacteria in wherever they left and brought it to Haiti and had it there for some time. What's the business about cat? What did we talk about last time that made him talk
Cat
about?
in the heart. The cat in the heart.
Cat in the heart.
Right.
Very good. I don't know about the CRISPR and modulating the phage population.
I'm not sure that CRISPR evolves that fast.
I think
Well-
that... Go ahead.
Right. So CRISPR is an acquired immunity system, so it can add sequences, and that's kind of the basic function going there. So yes, it can operate that way, but as Bill intuits here, the phages have their own responses to this, and so the bacteria never completely win and expunge the phages, because if they did, then there just wouldn't be any phages.
As to what's going on in the baby, I don't know. I mean, it may just be lysis of the population by phages. What the role of CRISPR remains to be found, right? Because nobody's looked at it there.
Yes.
It should also be pointed out that there are a slug of different ways that bacteria can be resistant to bacteriophage.
Sure.
Right. One easy one is one of the earliest studied, which is just attachment. Bacteria can alter their surface so that the phage can't attach anymore. And there's restriction modification. There's all kinds of stuff.
Dickson, can you take the next one?
Sure.
Please.
Dennis writes, Ducks, just a quick follow-up on the UVC robot for helping to clean contaminated rooms. I wondered whether UVC could kill viral particles. According to this Googled answer below, yes. Just a note. It will not be long before the UVC cart will become an increasingly capable robot. Elon Musk asserts that Teslas will fully self-drive from 2018 on. Daimler-owned Freightliner trucks are now legally self-driving across Nevada. I didn't know that.
I didn't know that either. Of course, in Nevada, you can just put any phage one.
Yeah, well, remind me to stay out of Nevada for a while until they've perfected it. Have you seen the Japanese robot prototype that can care for invalids? It can pick up a patient and gently move him or her around. In the not-distant future, this capability plus UVCs could become a cost-effective method of partial disinfection. Google answer, yes, germicidal UVC lamps kill up to 99.9% of most viruses, airborne bacteria, and mold spores. Yes, germicidal UVC lamps will kill up to 99.9% of mold and help prevent future mold growth. And then he gives an answer source for that. So thanks, Dennis.
I remember now the rationale behind using UVC in these robots, because UVC is filtered out from sunlight by our atmosphere. Only A and B get through, and so the idea is that the kind of damage caused by UVC, the organisms are not evolved to repair it as efficiently, and so it works better. Whether that's true or not, I don't know, but that was the rationale.
Yeah.
Well, and that, I know that they do. I think we were talking last time. Well, what makes this special? It must be the fact that it's a robot and it moves, because the food industry has used UVC for quite some time, and I think it was back in 2011, the FDA action to use this type of technology. I think there was a bunch of E. coli outbreaks linked to, it was unpasteurized apple cider or some type of fruit juice, contaminated fruit juice. And so then that put in place to use these UVC treatments as an acceptable method.
Yeah, I wonder if Toronto uses that, because they UV treat all of their effluent after they've used it.
Kathy, can you take the next one?
Sure. This is from Greg, who writes, Dear TWiV Illuminati, I'm writing with regard to the discussion that you had on 2 recent TWiV episodes, 353 and 354, concerning viruses bringing cellular molecules along for a ride. This began as a more specific discussion of the incorporation of cyclic GAMP into virus particles, where Vincent raised the interesting question, will anything that is overexpressed in a cell end up in a virus particle? On the following episode, Rich Condit mentioned a paper published in 1987 that looked at incorporation of a foreign protein into pox, which in that case could be detected by virtue of the protein's enzymatic activity. Ultimately, you came to the follow-up idea of expressing GFP during infection to see if fluorescence could be detected in virus particles. Well, the answer is assuredly yes, at least for a herpes virus. 2 years ago, I had a talented student in the lab, Kevin Bohannon, use the GFP, RFP, dual-tagged pseudorabies viruses to look at viral particle architecture. One of the many controls in this study was a virus that expressed GFP that was not fused to a virus protein. That is, upon infection of a cell, GFP was produced but not targeted to enter newly assembling virus particles. GFP was packaged into virus particles nevertheless. Although the level of GFP incorporation was variable from particle to particle, it could reach levels that were comparable to a lower abundance viral structural protein. Because GFP can non-specifically package into this enveloped virus, I would suspect something as small as cyclic GAMP would as well without the need of a directed mechanism. A danger of being enveloped, perhaps. Keep up the great work, and best to you and your co-stars, Greg. We can say his full name because he's a faculty member, Greg Smith. And he comments that it's a beautiful, sunny, 70 degrees Fahrenheit, 21.1 C Celsius day in downtown Chicago. And he gives a link to the paper describing the work.
Greg's at Northwestern, right?
Mm-hmm.
Yeah.
And he was on TWiV when I visited a long time ago.
Nice. So that's a pretty recent paper, too.
Yeah, so this should have been follow-up last week, but I missed his email. And because we had done these last week for the previous episode, so that's why it's now. In case anyone is wondering why it's a follow-up when it was 2 episodes ago, I'm sure there are some out there who are wondering.
Just say it's a slow virus.
And Steph, can you take the next one?
I sure can. Yeah, so last week on episode 355, you guys all talked about the infant gut virome. And so I really enjoyed the discussions of the milk
This is, by the way, Steph reading her own email.
Oh, I'm sorry.
Exactly.
I should probably preempt that, huh? Okay, so I wrote in, and I said, by the way, love this past TWiV 355. Naturally, I enjoyed the great discussions of the milk virome and how it contributes to the infant gut virome. So this idea I would like to maybe potentially work on in the future, because there really hasn't been much work done on the milk virome. There was some blog that talked about, I forget where he's from, South Dakota State, and now he's in San Diego, and they were doing milk virome research. We know that there are viruses that can actively replicate in the mammary gland, HIV being one, mouse mammary tumor virus being another, and then secondarily, there can be viruses that could end up in the milk. I had talked before how you have these cells that could, from the gut, let's say, traffic to the mammary gland, and if that virus replicates in B cells, then you may have a trafficking, replicating virus-containing B cell in the mammary gland that then contributes to that infant's virome. So there's really, though, not much information on non-HIV eukaryotic viruses and their contribution.
Are you also interested in the amniotic virome?
Yes, I would assume so. I know Dickson there was a lot of discussion. There definitely is an active microbiome in the amniotic fluid, but there's just not as much, and a lot of it is just little pieces. But
Steph, do you think there's any fluid in the human body that's sterile?
No, I do not.
Not even CSF?
Well, I mean, can't you have viruses in there too?
How about cochlear fluid? How about aqueous humor?
My fiance's in optometry school, and I always hound him on eye. You know what? There are immunological privileged sites of the eyeball, I'm just maybe that. But I would still say no.
What, didn't they find Ebola in the eyeball?
Yeah, they sure did.
Yes.
No, but that's not a normal virus.
Yeah, it sure isn't.
But there is also some evidence of a bacterial microbiome in the brain. I'm not sure how well
I
that's
read that
checked
too.
out.
It's in the tissue.
We did that on TWiM a long time ago.
Tissue.
There was one paper, and it's gotten a lot of criticism.
But all of the culture studies from, if you want to know whether it's a meningitis or not, you take CSF and you culture for everything, and you don't find it usually unless it is Neisseria. So,
right, but
what is not culturable that is there still?
Mm-hmm.
Exactly, there are a lot of things that are not culturable that you could. I mean,
I would agree.
When I was in college, they taught that these various environments that just couldn't be sterile were quote unquote sterile, meaning normally you can't culture anything from them. But anything, certainly anything that's contiguous with the outside world, the lungs, what have you, that's all going to be loaded with stuff.
I was quite amazed to see how many things live in the stomach, considering how harsh an environment that is.
Well, sure, but undersea volcanic vents are harsh environments.
No, right. So we we are a walking ecosystem,
there are,
no question about
pH 2 lakes where plenty of bacteria grow.
Yep.
No problem with extreme conditions. Nope. All right, we have 2 snippets for you today. One of them was sent by Steph, and it is a paper in PNAS gut DNA viromes of Malawian twins discordant for severe acute malnutrition. From a group at WashU and Columbia and Malawi and San Diego. The first author is Rise. And also on this paper, we should know David Wong, who was in last week's paper, Herb Virgin, Skip Virgin, Forrest Rower, and Jeff Gordon. And this is all about the virome of kids who are malnourished. Severe acute malnutrition. It's known that their gut microbiota isn't right, and this paper they wanted to look at the virome. And these kids who undergo severe acute malnutrition, this is something I learned from this. It can manifest itself as wasting, progressive wasting, which is called marasmus, or kwashiorkor, which is a syndrome including edema, hepatic steatosis, skin rashes, ulcerations, and anorexia. So they had done a study. Do you know
the difference nutritionally between the two?
No, I don't.
So kwashiorkor is really a lack of protein. And in fact, the name comes from potentially the Ga language, which is a language of Ghana, where it means the sickness the baby gets when the new baby comes. In other words, when the baby is weaned because there's another baby that comes along, then the weaned baby doesn't get as much protein. And that's distinguished from the marasmus, which is a total lack of energy intake.
Okay.
And kwashiorkor, these are the pictures you see of a little baby who's completely emaciated except for a totally bloated belly.
Yeah, because that's from the ascites.
That's the edema.
That's the ascites that develops.
So they've done studies on the microbiome of these kids, and they don't mature as it does as you age, and they have a younger appearing microbiome. And it doesn't get fixed when they give them food. So they have ready-to-use therapeutic food, which is a peanut-based food that they give them, and it doesn't always completely recover the microbiome. So they wanted to know if what's going on with the virome, because they had the samples from these twins that they'd taken, and so that's what's looked at in this paper. And interestingly, they have, and as part of this study, they had 317 twin pairs from 5 villages in Malawi. It's amazing. And in Malawi, when one of the twins gets sick from one of these severe acute malnutrition diseases, they feed both of them, so there's no stigma given to the sick one, right? They feed both kids. So that's an interesting pair that you have to study there. And
sort of a cultural control.
Yeah.
Yes.
That's very cool.
Right. So everything is the same about these two kids. They're twins, and one of them developed malnutrition. The other one didn't,
Right.
which for reasons that I think we're still not entirely clear on. And then they both get this RUTF, this ready-to-use therapeutic food intervention.
Yeah, and part of the reason they're looking at the virome is they're trying to figure out, can we explain why one of the twins gets sick and the other doesn't, right? Now, interestingly, what's been done before, they have fed fecal samples from these kids to germ-free mice, and you can reproduce weight loss and dysfunction in the gut to these animals, and that's important as part of this study.
And by the way, the RUTF is not very effective generally.
Yeah, it's not.
That's really kind of the core of what they're getting at, is why doesn't this work? The kid lacks protein. You load him up with a bunch of peanut butter. You're giving him plenty of protein. Why didn't you fix the problem?
Yeah.
And in those mouse studies, what's interesting is it is really completely reproducible in the mouse model when you have 2 different groups of mice, and one is fed a Malawian diet and one is fed a diet with higher protein and energy, and you put the bacterial gut, the fecal matter, in these mice. The ones that are fed the healthy diet still cannot gain weight. They cannot achieve the same type of weight gain and just normalcy. So there's this disturbance in the gut that is predisposed to, like what you mentioned, even trying to solve the problem with food additives.
So they have a subset of this original study, 8 monozygotic and 12 dizygotic twin pairs, 0 to 30 months of age in the 5 villages. 6 of the pairs had developed, one of the kids had developed kwashiorkor, and the other 6, one co-twin, developed marasmus. So they purify virus-like particles, and then they do amplification by multiple displacement amplification, which we talked about last time, and then they do deep sequencing. So they're just going to get the DNA viruses from this, and that's in fact what they find here. And they find a lot of the viruses we talked about last time, caudovirales, microviridae, phages of different kinds, alpavirinae, a new subfamily of phages with bacteroidetes, and anelloviridae, the very abundant viruses that we talked about last time, which they found in the other study. So what is the relationship between the viruses that you have and whether or not you are sick? So some of the interesting things that they find is that, well, as you would say, co-twins are more similar to each other among all of them. There are greater differences in the viromes between a twin and his or her mother or sibling and any two unrelated individuals. The similarities in the we're talking about the DNA viromes now, of course, of unrelated young twins are higher than between the twins of their mothers or older siblings. Age is an important driver in the virome composition. And they also make a comparison to that study of adult twin viromes that we talked about last time, which is they showed was highly distinctive and stable. And in contrast, in their Malawian study, there's much more similarity in the early life DNA virome.
Where they have results that you could compare to the baby gut virome paper that we talked about last week, they are pretty much complementary or the same. For instance, when they see the anelloviruses, they're highly abundant early up until about 15 or 18 months of age, after which time they diminish. They also see the same kind of progression from the one type of phages to another type of phages as the infants age. So I thought that was pretty nice that they got similar results.
So now if you try and say, can you distinguish...
So wait a minute, I want to make sure I got something straight here. Is it correct that in discordant pairs, that is one sick twin, one healthy twin, the virome is still is comparable?
Yes.
Right?
Mm-hmm.
So sickness and there is a change in virome that is basically correlates with illness. Okay? So you can identify a virome that looks like an ill virome. And yet you find that virome in both the healthy and the sick kid.
That's right. That's exactly right. It's exactly right. They're both twins. The virome is perturbed when you have
that's
one
a
of them
puzzle.
sick. That's a puzzle.
Anyway.
That's right. And what is perturbed, I think it was 30, I can't remember the number, but it's largely the eukaryotic DNA viruses is what is perturbed, which is quite interesting.
Yeah. So these twins are living together.
Yep.
So what about twins that are separated at birth?
I don't think they have that.
No, I'm sure they don't, but that would be the next group to look at to see where the divergence occurs and that sort of thing.
And then they look at the effect of feeding the RUTF, and there's really no difference on the virome as far as we can tell. So it's 37 of the disease pair discriminatory sequence groups, contigs, were anelloviridae or circoviridae. So that's higher than the other ones. Yeah. So it's quite interesting. And remember, last time we mentioned in their study, they used those as a marker of immune competency because they think that the less immune competent you are, the more diversity in these viruses that you're going to have.
So in the normal twin that's not affected by the disease that got this high protein diet as an intervention strategy, did they then gain weight better because they're not sick?
I don't know the answer to that.
Right. I don't think that's those data are not here.
Not part of this study. So then they took, this is what I think was pretty cool, they took some fecal samples from these kids and they fed them to gnotobiotic mice, and then they take the feces of those mice and they do some virome analysis, right? And remember, these mice will duplicate the disease, right, by being fed the feces of the sick kids. But none of the eukaryotic viruses come out of these mice. They don't replicate in them. They didn't detect any eukaryotic viruses in the fecal samples of these mice, again, who were fed fecal matter from the sick kids. So they think maybe these guys either are not involved in the disease or there's something else.
Well, the results that they get, the fact that you've got a healthy twin and a sick twin who have this similar microbiome dysfunction
Yes.
that is going on. So at the microbiome level, they both look like they ought to be sick, but only one is.
Mm-hmm.
And that suggests, at least to me, that the microbiome effect is not causative. That's actually an effect of whatever the underlying mechanism is.
You're talking about microbiome or virome?
Sorry, the virome.
Okay.
The virome that they're looking at, the dysfunctional virome that they see here, is more of an effect than a cause.
Yeah. Well, we don't know. I think that's
Maybe.
absolutely right.
I mean,
Yeah.
it could be bundled up, but there's something else going on, obviously, that is causing this.
Yeah.
So is the microbiome is also changed?
Mhm.
I mean,
Yes.
this is a question. The microbiome is also changed in the disease state, correct?
Correct.
Yes.
And if you transplant that into an animal, that animal expresses some of the disease manifestations, right?
Correct.
So is that microbiome also altered in a similar fashion in the healthy twin?
So from what I looked at a couple of their previous papers, and they do see a similar effect when discussing the microbiome, which, would like to see combining these two data and how they complexities of the microbiome and the virome. But yeah, and what's interesting is they really only say 7% of the cohort is concordant for undernourishment, meaning both twins are going to be undernourished. I just thought that was really surprising. Only 7%. That the majority of these twins only have one that experiences this SAM, or severe acute malnutrition.
So if you have a discordant pair, is the microbiome in both delayed? Do they both have a younger microbiome?
I'd have to go back and look at that paper, but that was the impression I got, was that they both do.
Because my next question was going to be, if that is the case, if you do the transplant of the microbiome from the healthy twin,
that's not going to help.
Does that, and stick it into mice, do the mice manifest disease? Okay, I'm looking for cause and effect here.
Right.
It gets complicated.
Yep.
Yeah, it doesn't mention here whether if you do that experiment, what happens. Yeah.
Right, but at any rate, in this particular case, all the data say that with respect to the virome, the proliferation of anelloviruses and et cetera are a marker for the disease state, but there's no evidence that it's causal.
Exactly.
Right.
Yeah, I'd love to know whether they're suffering from malabsorption syndrome also, and that's further up in the gut, in the small intestine, flattened villi, that sort of thing, that you could correlate with the malnutrition state that didn't happen in the twin. So to find that out, you'd have to biopsy, and that's just tough. Tough.
Mm-hmm. Steph, anything else we missed in this paper that you wanted to point out?
No, I think we covered a lot of it, although there was a lot of discussion in this paper on terms that I have not been trained on so far, and I think one of them was random forest regression.
Right, so I
which
looked
we
that
could
up.
talk about if someone knows more.
Well, I think we could just refer people to the link. Maybe
Yes.
Vincent can put that in the notes. That is a really good layperson's analogy. It's a algorithm for machine learning that they used on their data, for instance, to determine the chronological age of a healthy donor of a given fecal sample, whether that could be predicted based on the DNA virome. Anyway, so they use this random forest thing that has out-of-bag error rates. There was not an analogy to explain out-of-bag error rates, but yeah, it had some pretty fun terms.
The bioinformatics is getting it's getting so it's like reading a physics paper or something like that.
Right.
Yeah, so
It's
I have a comment on that, because actually I'm going to talk about readability probably of all the papers today, but compared to last week's one, I thought this one really suffered in readability. Maybe in part because it's PNAS, so it was constrained, but the second and third last paragraphs in the whole paper have one sentence paragraphs, one that's 9 lines long, one that's 11 lines long. And then in the last paragraph has another 11-line long sentence. somebody's got to give these people some punctuation by a period or.
Yeah. So there's in forestry, I just wanted to add a note here. You can actually tell the age of a forest by taking random samples of trees and then seeing how big the forest is, and then from that you can actually tell what the age structure for the trees are for the whole forest. So it's a way of taking a small sample and connecting it to the larger samples. And so,
well, in this random forest, I gather it's because in order to do this machine learning algorithm, you build something called a decision tree. And then the random forest is a bunch of decision trees, each configured a little bit differently to kind of average out an answer.
All right, we have another snippet, and this one was sent to us by Rich via Grant McFadden, and it's a paper in PLOS Genetics. And Grant just had 3 words. I love this. Recurrent domestication by Lepidoptera of genes from their parasites mediated by brachoviruses. And this is from Spain, France, and France. Yeah, Spain and France. The first author is Leila Ghazmi, and the last author is Jean-Michel Drezin. And you know, Rich Condit, you love this wasp stuff, don't you?
I love this wasp stuff.
Why don't you tell us about this?
Okay, so I got to back up and do the polydnavirus thing. I will
remember,
try
this
and be
is a
brief.
snippet. It's a snippet, right?
I know. I will try and be brief, but it's complicated. So,
watch out when they say that.
This concerns parasitoid wasps. These are wasps that lay their eggs in caterpillars, and the caterpillar host provides an environment to nourish the eggs where they hatch as larvae. And the wasps, during the egg development, elaborate this virus in the oviduct that is laid along with the eggs. It's called a virus because it's got virus particles, really nice icosahedral particles sometimes, depending on the virus, and they're full of a bunch of different circular DNAs. There can be 50 of these things that are different sizes and encode different genes.
And we did this in one of our 2014 shows, didn't we?
Yep.
Yeah.
We've done polydnaviruses a couple of times. And when these viruses are delivered along with the eggs, they infect the cells of the caterpillar that the eggs are laid in, and the circular DNAs in the capsids are delivered to the caterpillar cells, where they then integrate into the cellular DNA and make proteins that are immunosuppressive to the advantage of the eggs. Okay? Now, there's no virus replication or anything that goes on. And there's a set of genes that all these genes actually come from the wasp genome. Okay? And during the egg development, they're amplified out of the genome and packaged into these capsids and delivered. But there are no replication proteins or anything else. There's another set of genes in the wasp genome that in fact are evolved from some ancient nudibvirus infection, another DNA virus that can infect and sometimes integrate. Apparently that happened over 100 million years ago, and that supplies genes for stuff like capsid and transcription of viral genes to get the viral genes or the nudiviral ancestral genes going, and that's part of what helps amplify this other set of cellular genes that get delivered. Otherwise, those virus particles that deliver the circular DNAs, they're just gene delivery particles. There's no virus replication going on.
At some point way back in evolution, these wasps were infected with a couple of different viruses, and instead of succumbing to those viruses, they stole them and used them for this parasitoid life cycle.
Right, and
weaponized them.
Right. So, and the viruses that were integrated, that were stolen, provide genes that help amplify another bunch of genes that have these immunosuppressive effects and are delivered to the host. In this paper, there's a good figure 10, that nicely summarizes these two sets of genes and summarizes the paper. It's really
And this
well
is
done.
an open access paper, so you can all go look at it.
Right. This is conceptually really tough because
Yes.
it's outside of the normal realm of things,
Pretty
and it's hard to
remarkable.
describe.
It's amazing.
Okay, so you want me to go on with the paper?
I do.
I mean, the
pretty-
fundamental observation that they've made by sequencing, by genome sequencing of a bunch of, or mining sequences of a bunch of different lepidopteran hosts, is that they find that some Lepidoptera who are not normal hosts of the parasitic wasp, and in fact some that are, contain sequences that represent integrations of these circular DNAs, we're going to call proviral DNAs. Circular DNAs that are delivered with the virus. And they can even tell that they actually came from the virus because sometimes they contain not only the genes, but also signals that are flank the genes that are associated, that are peculiar to the proviral sequences and not the wasp genome, that prove that they are viral in nature or were originally viral in nature. So what they're thinking is that there's a couple of different scenarios. One scenario is that although there's a high species specificity between the parasitoid wasp and a host, occasionally they get lazy or they miss or something like that, and they go and infect the wrong lepidopteran. A lepidopteran who, for some other reason, may be naturally non-permissive for the growth of the larvae in the caterpillar. And then the effect of laying the virus in that non-permissive host is that the DNAs get integrated, and some of them get integrated in the germline. Okay, and so now you've established a line of Lepidoptera that contain some of these genes that were delivered by the virus. They also suggest that occasionally the normal host can, for some reason, survive, and the same thing can happen. Some of the genes can be integrated into the germline and then passed on. So the bottom line is that this virus delivers genes that ordinarily are for an immunosuppressive function, but can also, on an evolutionary scale, serve as horizontal gene transfer. So they're transferring these genes to various different hosts.
So this is horizontal gene transfer from one animal to another species.
Via a virus, yes.
Via a virus.
And even across orders.
Yeah.
Yes.
Yes.
And they, at one point, this is a cool part. It's all cool, but this is cool. They try to produce some of these proteins in baculovirus vectors, because that's a standard way, and they find that two of the proteins actually inhibit baculo replication. So and these are pathogens of the caterpillars, right? So it looks like not only have these genes been acquired from wasps, but they have a function, a protective function. It's amazing.
Against a completely unrelated virus.
It's
Yes.
gain of function. We need to tell these guys to cut it out.
So I was going to ask, a larval wasp developing in the hemolymph of a caterpillar is a pretty big object. So
Yes.
what portion of the innate immune response system for that insect is this virus inhibiting?
I don't know. First of all, I don't know any insect immunity, and second, I don't know the functions. As a matter of fact, the functions of a lot of these genes are not well understood. But my guess is that it's multiple, many different facets of the immune system, because we're talking about at least 50 genes in many cases.
Oh, wow.
Okay,
Okay.
so there's an opportunity here to compromise several different immune functions.
Most of the insect immunity is by encapsulation with hemocytes, and then it creates a capsule around something, and then it creates an unhealthy environment. The organism then dies inside the capsule, and you get like a cyst formed, which has no life form inside of it, and that's how they usually take care of their, large parasites like nematodes and things like that. But
Right, so I don't even know in this case.
It
I don't even know what.
was an you wouldn't call it mitotic, but it's an anti-DNA. Maybe it prevents the bacteria from replicating at the DNA level. That'd be very interesting to know about, because that's a useful substance for some other bacteria.
So, at any rate, I don't even know what the physiology of the immune response is in this case, where the virus is, what happens to the hemocytes and et cetera.
So it sounds like you could do that experiment, though, couldn't you?
Sure. Good.
Yeah, sure. Get funded, you could do it, Dickson.
Just knock out that circular DNA.
I'm so happy this kind of stuff gets done, You could
absolutely. It looks esoteric, but look at, if you discovered that the baculovirus was inhibited because its DNA replication cycle was interrupted, maybe it's a useful substance for TB.
So they go on in their discussion to point out other, I didn't highlight this, so I don't know if I can recover it, other examples of virus-mediated horizontal gene transfer. Of course, the one we talk about all the time is retroviruses in the syncytin gene that is important for placental mammals.
Of course, phages do it all the time. Phages and bacterial hosts.
Right.
Both ways. All right. Now we have a paper.
Wait.
Yes.
I just have a comment. This is, in terms of readability, it was a bit of a challenge because it's a very genetic paper. It's in PLOS Genetics. And Vincent has written a nice blog post about this that readers might, or listeners might want to take a look at. And I just had a comment about the title, Recurrent Domestication by Lepidoptera of genes from their parasites mediated by brachyviruses. To me, this is not the right use of the word domestication, and that really what they mean is something more like recurrent integration or recurrent endogenization or something like that. But domestication is adapting wild plants and animals for human use. So
this term has started creeping into several areas of molecular biology. I heard a talk, CRISPR talk, which used this because the CRISPR system itself it borrows pieces from bacteriophages in order to carry out its function. And the speaker suggested that this was the domestication of the phage.
So we can now in the future interrogate the domestication of genes, right?
In fact, you can interrogate the phagome by the domestication of the phage. So this is a borrowing of a term. I think somebody initially did it to be clever, and now it's spreading. I don't have a strong opinion of it yet, but I may get sick of it soon.
That's why I was gonna bring up interrogation, Rich. You did it. All right, can we do our paper now?
Sure.
Yes.
All right, our paper was suggested by Stephanie. So Stephanie, why don't you tell us all about it?
Yeah, sure. So the paper title is Chemokine Adjuvanted Electroporated DNA Vaccines Induces Substantial Protection from Simian Immunodeficiency Virus Vaginal Challenge. And this paper was by the last name Kunstler and Wise, and it does tell you in the paper that both of those authors contributed equally, although I don't think I would like to be the person with the W last name, because unfortunately we are only going to call it Kunstler et al. And not Kunstler and Wise. So, and they're from Drexel University College of Medicine and University of Pennsylvania. And I chose this paper for a couple reasons. I think it brings into the conversation things about DNA vaccines. We're going to talk about mucosal immunology and chemokine-driven lymphocyte chemotaxis. And also, there are some interesting ways they present the data here, and we'll get into that. So what they begin by talking about is this paper highlights the current need for an HIV vaccine. And while I am more of a mucosal immunologist, coronavirologist, I'm not an HIV virologist. So for the HIV virologists out there, you can always write in and correct me. But from my understanding, there have been multiple candidates for vaccines for HIV. I think one of the trials that received particular attention was that RV144 trial. They mentioned this in the paper. And that was done in Thailand. And they utilized this priming vaccine and then subsequent boosting regimen with these two vaccines. One of them consisted of a recombinant canary pox viral vector that expressed the envelope, the nucleic capsid, and the polymerase proteins. And then the second one was, I think, it was a GP120 surface protein. A recombinant surface protein vaccine. But that trial only resulted in 31% vaccine efficacy. So it's only a modest rate of protection, and it really didn't last. There wasn't subsequent protection over time. So that study, while it was only modest, it did kind of, reinvigorate HIV vaccine research, because as we know right now, there's only antiretroviral therapies to treat HIV. So this paper, what they wanted to do is they wanted to focus on DNA-based vaccines. So the fundamental idea behind a DNA vaccine is really to induce immune responses against recombinant antigens that are encoded by engineered DNA plasmids, but they're actually going to be expressed in vivo in an animal model or in the patient. So what happens is there is a plasmid that has a DNA, an engineered DNA for whatever antigen you would like to express. So for instance, in our paper, they use a plasmid that expresses the 3 HIV proteins, GAG, POL, and the envelope. And then what that's going to do is if you inject it intramuscularly, 2 things. The myocytes, or the muscle cells, are actually going to uptake that plasmid and are going to use their own host cellular machinery to facilitate the expression of the plasmid encoded gene. And then that's going to lead to the generation of foreign antigens, which then will be processed and presented by antigen-presenting cells, dendritic cells, macrophages, and some B cells. So these host-synthesized foreign antigens are recognized by the immune system and hopefully would then confer complete or effective immunization. DNA vaccines have showed success in animal models, but what really hasn't been true is when we've used them in humans. There has been a cellular immune response, so a CD4 or CD8 antigen vaccine-specific immune response, but the humoral immune response, the B cells that then differentiate into plasmablasts to secrete IgA, IgG, that humoral immune response has not been shown to be as effective in DNA vaccines in humans. So knowing that there is a need to improve DNA vaccines, and particularly in HIV research, there have been efforts to include other adjuvants. IL-12 has been used in, I think there was a trial, it was like, I think there was a HIV trial 8080, and that used IL-12. IL-12 helps to stimulate the memory T cell response, particularly CD4 T cells, which we know would be important in HIV. The second thing they used was something called electroporation. Electroporation is something they also use in this paper, and what it is, it's a method to introduce macromolecules such as nucleic acids, or in our case, a plasmid, into the cells, either in vivo or in vitro on the bench. And it's just brief electric pulses to induce a permeabilization of the cell of the lipid membrane. So what they did in this study was they used these 2 methods. So they did, the treatments included rhesus, I'll go back and talk about the animals that they used. They did use rhesus macaques. They used Indian rhesus macaques, which are susceptible to simian immunodeficiency virus. And this SIV is used a lot in HIV vaccine research because it does confirm a type of simian AIDS over time. So they used I think it was at Tulane, they used a cohort of rhesus macaques, and they then wanted to vaccinate them, and they used, to vaccinate them, they used a plasmid that then encoded the 3 proteins I mentioned before, the HIV envelope, the HIV nucleocapsid, and then the HIV polymerase. And then they also, in addition, they vaccinated a second subset of rhesus macaques with CCL, a recombinant, CCL25, and then recombinant CCL 27 and 28.
So I wanted to ask you, actually, it was a good point. How were they immunized? Where are these IM injections with these?
Yes, so they are we recording? I can keep going.
Yeah.
Okay, yeah, so they're IM injections, and what they say in the methods is they gave IM injections in 2 different spots, which they don't tell you where the spots were. That would be really key for me. I'd like to know where, if it was on the leg close to where HIV infects, which would be the vaginal tract, or was it in the arm? They don't really say, but what they do say is after the injections of the vaccine, they gave an electric pulse with forget the name of the company, but it's basically just a electroporation machine, and they use that directly after.
The company is Inovio.
Inovio, yes.
Yeah,
right.
And they've got a machine that does this, and it has there's a probe with a couple of needles on the end of it that I assume deliver the DNA for the vaccine, but they also serve as electrodes.
Yes.
That give one's positively charged, one's negatively charged, and they give a pulse, a charge pulse at the same time. So you get the electroporation and the delivery of the DNA simultaneously.
Does this hurt?
Well, I mean, unfortunately, rhesus macaques can't talk, but I don't know if it would hurt or not.
Bite.
They do. But now they have done this method with, there's 4 DNA not necessarily vaccines, but therapeutics, and they're mostly used in veterinary medicine. I think there's a West Nile one. There's growth hormone in pigs, and they use this electroporation. But in humans, I'm not, to be honest, sure. So
who's the West Nile one for?
Horses.
Horses. Yep.
And the horses don't talk either, except Mr. Ed.
Actually, you beat me to that one.
I'm just curious about, if this really works. It must have been tried in people. If it really is painful, it's not going to be accepted very well.
Right. I would assume they have done it in people, and I would with gene gun therapy. I mean, it's not electric pulses, but you do have pressure there, so you would just, I guess.
I can't imagine this being so much of a charge that you would actually hurt from it.
No, I don't think so. So actually, I may be mistaken here, because they say they deliver the DNA in 2 separate sites followed by in vivo electroporation. So it could be that the injection and the electroporation pulse are 2 separate processes. Yeah.
That's the way they describe it in the paper. Yeah, suggests that it wasn't at the same time, which I did look up where the company, this electroporation machine, and it looked like you could actually vaccinate and give the electroporation at the same time.
That's what it looked like to me.
Well, they don't say how many milliseconds after.
Right.
So it may be quote unquote simultaneous, but actually sequential.
Right. So I want to talk a little bit about the adjuvants they used in this vaccine, because they do relate to mucosal immunology and the trafficking of cells to the site of infection, or in this case, the site of the vaccine. So they used CCL25, CCL28, and CCL27, and that just stands for chemokine ligand. This chemokine ligand is something that is soluble, that is secreted from a particular immune cell in the mucosal environment. So, like, for instance, CCL28 is constitutively expressed in the small and large intestine. This expression can be enhanced during infection, and they think a lot of the secretion of these intestinal chemokines are then stimulated by the microbiome. So there is an interaction there. For CCL25, it's mostly expressed in the thymus, which helps to stimulate the migration of T cells to that spot. And then for CCL27, it's a skin homing chemokine that's secreted by skin cells to aid in the migration of T lymphocytes in the skin. So the way they present this paper, they vaccinate these animals, but they then present us data referring to them as CCR9L groups and CCR10L or ligand groups.
Right. So
what that means is these chemokines are being secreted by cells. They're then binding to, let's say, a B cell, G protein coupled receptor on the surface of that cell. So once it's bound to CCR9, which would be a receptor for CCL25, it then induces this cell to release, I think, calcium, and this then is a chemoattractant for the cells to start moving. And the closer and closer the cell gets to the chemoattractant, the higher expression of CCR9 or CCR10. But what they did here, because it can get a little confusing, they treated the animals with these CCL 25, 27, 28 individually. But they combined the data presenting as one group was CCL25 vaccinated as an adjuvant, and then one group is CCL28 and 27. And we'll go through the results, and I'll kind of explain why I think they did that. But for the setup, did I do a good job of explaining, or do I need to?
Yep,
That's okay.
No, that's good.
That's good.
Okay, perfect. So again, the general point is we want to see if this DNA vaccine adjuvated with chemokines and electroporation can help in vaccine efficacy. So the first figure that they give us is demonstrating what the vaccine cellular response is. And really they focus on interferon gamma. Interferon gamma is secreted from CD8 cells and also from CD4 cells that have differentiated after being presented with an antigen from dendritic cells. In the first ABC figure 1, they just do a time course looking at the amount of interferon secreting cells and then normalize to the total amount of cells that they recovered from that animal. And what they demonstrate is that CCR9 adjuvated and vaccinated animals have higher amounts of interferon gamma. And that happens after week 20.
This is serum, right?
And this is in serum, that's correct.
This is... just so you know, the listeners will, by and large, not be able to view this paper.
Right.
This is closed access, very closed access, apparently.
Yeah, even we couldn't get it. Yeah,
Well,
I used my Nature press credentials to grab the paper.
Oh, my goodness. Well, yeah, so this one, I guess Ohio State subscribes to mucosal immunology, because it is a Nature press.
It's a Nature journal, but it's one of their smaller journals, so it's not, if you get Nature, you don't necessarily get access.
Yeah, I had to get it through interlibrary loan. I got it within a
good.
day.
So
but,
maybe,
yeah.
Stephanie, you could just summarize the figure rather than referring to the panels.
Oh, sure. So the summary, what they're really trying to look for are how did this vaccine induce the immune response and what kind of correlates can we take away from that. And the animals that were adjuvated with CCR9 the ligand, which would be the CCL25 chemokine, they had higher amount of interferon gamma secreting cells. And CCR10 ligand adjuvated, they had a substantial, they call it a substantial increase, but it's actually not significantly different. So one of the things about this paper is that they do refer to things as being substantially increased while there's really no significant differences.
That's why they don't say significantly increased.
Exactly. Yeah.
So, Steph, the interferon gamma is used as a surrogate for T cells. Is that right?
That's correct. So, and when you're looking at cellular immune response and you're looking at lymphocytes, CD8 and CD4 cells that have been activated, interferon gamma is something that you would look at for a memory response. Okay. They go on then, they want to look at these certain cytokines that these cells produce. So they focus on IL-2, TNF-alpha, and interferon gamma, and they call these polyfunctional T cells and CD8 and CD4 cells. I think in the past that these polyfunctional cells, if they can secrete all three of these antibodies, they have been correlated to protection against HIV, which makes sense why they would look at it. But really, they don't find anything significantly different in terms of cells that can secrete all three of these cytokines. So really what we can take away from what these vaccines do to the cellular immune response is just higher interferon gamma secreting cells.
Let me interrupt you because Dickson has to leave.
I have to go somewhere,
sorry.
so it's nothing. It doesn't reflect badly on anybody at this show. I just had a pressing engagement that I can't postpone.
Do you want to give us your quick pick?
I have two of them actually. One is a colored figures from NASA for the Pluto flyby, which is just spectacular.
Cool. Yeah, those are beautiful.
And the other one is a prediction for next Monday to go out real early and see the blood moon. It's a remarkable eclipse phenomenon, which may not occur again until 2033. And it's worth trying to see it because I think it's spectacular. If the moon is as close to the Earth as it will be, and it looks 14% bigger than it ordinarily does.
Cool.
Yeah,
so
these
those
are both really
picks.
cool.
Those are my both picks.
Thank you, Dixon.
Nothing
Thank
about
you, Dickson.
Hey, you're
Bye.
welcome. I'm sorry.
See you next
about
week.
it. You betcha.
Bye-bye.
Okay, bye-bye.
Have a good weekend.
You too, everybody.
All right, go ahead, Steph. Sorry.
Okay. No, that's fine. That's fine. So, yeah, as I mentioned, what we can take away, cellular immune response wasn't substantially increased in any of the vaccines. You see some increase, but nothing significant. So they move on to, okay, let's look at the B cells. B cells secrete antibodies, IgA and IgG. IgA being particularly important at mucosal sites, and IgG being more important or more common in systemic immunity in the blood. So they, when looking at the vaccinated animals, they wanted to determine basically how many HIV-specific antibodies there were in the different vaccinated animals. So they show that there were higher amounts of serum IgA and IgG in animals that were vaccinated with CCR10 ligand adjuvanted vaccines. And so what that particularly means is they go on in this figure, they were trying to see if what particular protein that these antibody responses were specific to. Because the name of the game when we're really thinking about HIV vaccines and many other vaccines is broadly neutralizing antibodies, antibodies that can bind one or multiple proteins that then could mean that they could bind more than one of the HIV strains, which we know is a problem for HIV infection. So they show that P27, which I think is the GAG, yes, that would be the nucleic capsid protein, and then GP160, which is the envelope protein, there's higher amounts of antibodies specific to those specific proteins in the animals that were adjuvanted with CCR10.
So 2 major structural proteins.
Absolutely, 2 major structural proteins. And so what I take away from that and how I think they don't really discuss the mechanisms maybe of how these antibodies are being increased in animals adjuvanted with this chemokine. But when I think about the amount of B cells and plasma cells that are circulating in our blood, a lot of them are already have been upregulated either in the gut, in the vagina, in any of these mucosal sites. Their CCR9 or CCR10 could be already upregulated or hasn't been upregulated yet. So by adjuvanting this chemokine, you're essentially drawing those cells into the site of where you vaccinated. So if you're drawing in those cells, there's a higher likelihood that a dendritic cell or an antigen-presenting cell could present these major structural proteins then to the cells to generate a humoral response. So I think that what they show here as a significant enhancement is that the circulating B cells were able to migrate towards that site of immunization. And potentially could be a correlate of immunity when looking forward, which we'll talk about in a little bit. So does anybody have any thoughts or comments on just looking at the responses to the vaccines?
So the other thing, maybe you're going to get to this, but the other thing they measure is neutralization activity in a pseudotyped assay of serum. And that really, if I remember correctly, the adjuvants really gave a significant boost of neutralizing activity.
Right. So
which would be a manifestation of the antibody response.
Yes. So looking, they can't look at the paper. I apologize. Yes. So the serum neutralizing antibodies, when we do that assay with our work, we do basically like an opposite of a plaque assay. We have cells that we infect with the virus, and then we add serum. And if we add virus and then we add serum, and if you have less amount of plaques or less amount of places where the virus has really bore a hole into your well, if you have less of that, that means you have more antibodies that were able to neutralize. And they do show in this in the results here that the animals that the rhesus macaques that were vaccinated with the CCR10 adjuvant and the CCR9, they do show, again, there's a trend, but it's not significant. So maybe in the text, they do a little over-exaggeration of the effect of CCR9, CCR10. But what I can take away from that is that there could be a correlate of serum IgA and IgG to these specific HIV structural proteins that we could look at as potentially protecting these animals.
So the inhibition assay is sort of reverse virology?
Yeah, sorry,
I just had to throw that in there for Vincent.
Vincent didn't respond, so I was wondering where he was.
Yeah. So what I was remembering is that
He's just
they
stupid,
get-
right?
I'm here. Yeah, I just
what I remember is that they get a significant increase in the virus neutralization for all three relative to the naive controls. But
Yes.
right, there's no difference between the adjuvanted guys and the non-adjuvanted guys.
Right. Yeah, you do. There's a couple animals that were able to have an increased amount of production of these neutralizing antibodies, but yes, overall the vaccine increased that over the naive or just animals. I think they said for the control, they just gave them water and then the electroporation, which I think a more appropriate control would have been just a plasmid that does not encode.
So
Right.
give me a blanket summary. If you compare the adjuvanted with non-adjuvanted, what the overall IgG versus IgA, how do they differ?
So overall, there's an increased amount of HIV-specific antibodies in the serum in the animals that were adjuvanted with the chemokines.
And is that is across IgG and IgA?
Yeah, both of them.
Both go up?
Correct.
Okay,
Correct.
got it.
Yep. So they went on from there and they said, okay, well, those were the cellular and humoral responses. Again, we're seeing more of a humoral response in this paper. And they then want to know, well, we're going to challenge the animals with SIV and we're going to see how they respond. So they challenge them, I think, it's like a TCID 50, which it's a tissue culture infective dose is basically 50% of infected cells, and they took that number and they multiplied it by 500, and then that was the dose that they gave the monkeys. And they did a vaginal challenge. So at this point, they then measure the viral titers in both the I think we're moving on to figure 3. They look at the viral load in each of these different groups. So animals that were vaccinated with DNA only or the chemokine adjuvants. And they did demonstrate, so looking at these figures, they combined all the animals that were vaccinated with the CCR10 adjuvant and they show, I think, 88.9% protection. There was only one animal that was not able to clear the HIV infection. But when you look at the other animals, there were, let's say, 2 animals in the CCR9 vaccinated animals that could not clear the virus. And then in the DNA vaccinated alone animals, there was, I think, 3 out of 5 that were not able to clear. So they then went on to say, okay, well, if we have CCR10 ligand vaccinated animals that were able to clear HIV infection better, let's think about correlates of protection. Can we then take what we learned from the humoral response and then correlate that to, are these animals getting protected? So before I keep going, any comments or questions?
Well, yeah. So to me, one of the things that's interesting, and we can come back to this at the end as well, is that they see 2 types of protection. And this is going to be important for the correlates of protection as well. They see 2 types of protection. And this is as measured by looking for viral genomes in the blood. One pattern is that they never see any virus.
Mm-hmm.
Right.
And the other pattern, and I think they call that uninfected. And the other pattern they call abortive, where they see a burst of viral RNA in the serum for a little while, and then it goes away, and then for months afterwards, and they took the experiments out to 6 months, you see no virus. So to me, those are 2 very distinct patterns. One is you never see any signs of the infection. The other, the infection starts and then it goes away.
Right. And so that the next part, when they really go into it, so they show you that, CCR10 specifically being the adjuvant that was able to decrease the peak viral load, and how long that these animals, how long it took for them to be able to control the virus, assuming that these antibodies are binding to the virus and neutralizing them so they cannot enter the cells to replicate. They want to look at those 3 types of infections, as Rich mentioned. So they want to look at uninfected, aborted, and then progressive. But because that this study wasn't designed to split these animals up into these three different infection groups, they don't see any differences between the adjuvants, and they just move on to talk about what does the immune response look like, categorizing these three very specific infections.
Basically, the way the experiments fell out, they don't have enough animals in each of those groups to get a statistically significant answer.
Yeah.
Exactly. And so when working with larger animals, and what I would like to see them do with this study is, since they already are combining data from the CCL27 adjuvanted and CCL28 adjuvanted, they didn't give those animals both those chemokines at the same time. So if they could take this data and then take animals and vaccinate them with this DNA vaccine with both of those chemokines, maybe they would see an increase in effect, rather than just combining data from two different treatments. So yeah, at the end, they just don't have enough animals to be able to compare in these specific categories of infection, being uninfected, abortive, and progressive. Now, one of the things that I had to get into, since I'm not an HIV virologist, is this idea of an abortive infection. I think Rich had some questions about that as well in the show notes. We were discussing that. And
because first of all, I want to highlight why this is puzzling, in particular for listeners who are not, have retroviruses at their fingertips. So, at least the way it's ordinarily presented, retrovirus is an RNA virus genome that's delivered to the cell and reverse transcribed. That is, a DNA copy is made, and then that DNA copy integrates into the genome, and then that's transcribed into new virus genomes. So the integration into the DNA genome, now you got a permanent copy of the virus genome in the host cell, and that is an obligatory part of the infection. And to me, one of the issues, that's one of the reasons that it's always been so problematic to cure, or in particular cure, or even vaccinate against a retrovirus infection, is that if the virus replicates at all, then according to my understanding, you now have an integrated genome, and you're not going to be able to get rid of that. So under those circumstances, first of all, to me that implies that in the animals that were uninfected, that to me suggests that you're getting what I would call sterilizing immunity. Okay, the animals are primed with an immune response so that when they're infected with a retrovirus, the retrovirus never replicates at all. So you never see anything. It must be sterilized before it has a chance to do anything. But in the abortive infection, how do you do that? Okay, if you go through part of the viral life cycle, how can you get rid of the cells that have the provirus? Now, Stephanie, I think you have an explanation for that.
Well, yeah. So the abortive infection, it was saying that the RNA is reverse transcribed, but it's not integrated into the host DNA. So whatever cellular mechanism in that cell is able to you have reverse transcription, but you just don't have integration.
But it could be that it's so they never looked for proviral DNA, did they? Right. So that's what they need to do, and it could be that the levels are below detection, because we know that in chronic, elite controllers, often the proviral DNA is there, but the blood levels are so low you don't pick it up by highly sensitive PCR, and I bet their PCR is not all that sensitive here. So I'll bet they have integrated copies, and they're just not making a lot of virus. Right.
And which, so going on to the kind of what I consider the second part of this paper is when they're looking at correlates of protection for these three different types of infections, it does make sense then that you see abortive infections still have an increase in both vaginal and serum HIV IgA antibodies. And so when you look at these three different types of infections, the uninfected animals, most likely they don't have high levels of IgG and IgA. Now, they looked at, these were taken 2 months post-challenge. So one of the things I'm thinking when I think think about trafficking of cells, I do think that they're missing out on. They should be sampling these animals and collecting the cells in the antibodies before 2 months, because we know that stimulation at the local site, in this case the vaginal tract, it takes about 8 to 10 days for those cells to meet the antigen in the lymph node. I'm thinking in terms of a gut, so mesenteric lymph node, but the lymph node in the vaginal tract, to then replicate, to then enter the bloodstream, differentiate into plasmablasts, and then start secreting antibodies. So almost looking at 2 months post, those animals that are uninfected, they've cleared the virus. So they don't have a consistent stimulation and production of IgA. So that's why uninfected animals, you just don't see a lot of these antibodies. But in the abortive and the progressive, you see much higher levels of IgA and IgG, because those animals cannot clear that virus. And so you see a constant stimulation and production of antibodies. So that's what I took away from looking at I'm looking at figure 5, these 3 different types of infection.
So, Vincent, if you're postulating that the abortively infected animals do have cells that contain proviral DNA, then I take away from that the immune response is good enough so that even though there are a few of those cells around producing virus, that virus is constantly being knocked back. Is that the idea?
Yeah, but you would rather have a sterilizing immunity, right?
You want to get rid of it.
Because right now the problem we have is everybody on triple therapy still has genomes, right? And then if you stop, so you can imagine if you got immunosuppressed for some reason, these individuals who underwent abortive infection, if you get immunosuppressed, which can happen, you're going to get an ongoing infection again.
Actually, that would be a good way to determine in this model whether those animals are actually sterilized.
You could, or you just take some blood and make DNA and make genomic DNA and look for the provirus.
Look for the provirus.
Yeah. So that's what I would worry about there, yeah.
And in my mind, the reason that you didn't get a boost of a humoral response in the animals that looked uninfected is you don't get any virus replication, so you're not boosting the immune response.
Right. Those animals have already been able to, if they're able to clear that virus and have this, what we're calling sterilizing immunity, then there's no virus to, yes, like you said, stimulate to produce antibodies. And what I would posit is that you have virus at the vaginal site. Well, if you have, and going back to this idea of using chemokines, there were some trends. And so animals that can clear, once that virus is recognized by immune cells in the vaginal intestinal tract, it's going to sample that, and then it's that vaginal tract that will just start to secrete chemokines itself. And if there are circulating plasmablasts specific for HIV in blood, that then they're circulating, they're waiting for that moment when they get the signal, it's time. If the vaginal tract is then secreting CCL28, that vaccine was able to enhance these CCR10 positive cells to migrate to the site of infection, clear the virus, and then that animal has been uninfected, essentially.
So is this worth pursuing CCR10, et cetera? Or is this kind of the end?
Well, based on this paper, I mean, they just need more animals. I would venture to say that it's worth trying if you're combining multiple chemokines. But the problem is those chemokines, depending on where you're vaccinating, I don't know what the reach is going to be. You have to maybe have chemokines are constitutively expressed, and especially the site of infection, they're at much higher levels than maybe what, I guess they did do multiple vaccines, but I just don't think it's enough to be able to stimulate enough B cells to produce antibodies.
So I have a fundamental misunderstanding. So flu vaccines, some of them you inject, right? And they give you protection at the respiratory surface. But polio inactivated vaccine does not protect your gut. So what determines if an injected vaccine will stimulate homing of the right cells to mucosal surfaces?
Well, but the oral polio does confer.
Yeah, the oral is fine, but I'm talking about the inactivated, which doesn't confer, yet the injected flu does confer protection. What's the difference?
Well, probably whatever the conformation of that antigen is and how it is recognized by the cells could then be a reason why you just have a cell that it's going to bind tighter, it's going to recognize better to produce. So in influenza, to produce enough antibodies to then protect. But in your example with polio, it's never the inactivated is never going to stimulate enough in the gut, which is where that site of infection is in the gut. So that's why the oral is able to protect, because it's a live virus.
Right. But why does flu protect in the lung and it's injected as well? You're saying it's the kind of antigen that's being used?
I think that's one of the reasons, right? And it could be the dose that's given.
Are these IgGs that are going across, or IgAs?
So IgA, we know secretory IgA is really important at mucosal sites, but it could just be, I'm thinking back to flu, that IgG could just be more effective at the site of the lung, whereas we know IgA is the antibody you need in the gut.
Right.
We
We're
know
dealing
that.
with two very different compartments.
So you're saying that in the gut, IgG is not going to help you?
It's not that it's not going to help you, but it's not going to be as effective because it's just not producing the amount of quantities that IgA is. So I think if I'm remembering, for every kilogram, every pound kilogram per person, you produce 0.6. Oh, I had this number. Basically, I produce, as a 140-pound person, I produce about 4 grams of IgA a day, which is insane. So really at the gut, we know it's IgA. And that kind of goes back to this fundamental question of this common mucosal immune system. For a while, we said, well, if you vaccinate, in one mucosal site, you'll get protection in another because of this commonalities. But really, it's actually pretty organ specific, and it's a give and take between, we have these common chemokines receptors that I've been talking about, but it is specific to whatever organ.
But we know that flu mist is really good because you're infecting the mucosal surface. Presumably you get IgA produced. Why do this experiment? Why not do vaginal immunization, right? Because it would seem to be the most effective in theory, right?
It would absolutely be most effective. I just assume that they're trying to do what would be most possible in the field. I don't think that vaginal, because I think they're really trying to get at this DNA vaccine, which we know is beneficial, and DNA. I don't know how you could electroporate and then also intervaginally.
Or intrarectal. But I mean, that's nobody's going to do that, right?
No.
Okay, got it. I got it. Okay.
So if I could leave somebody with, leave the audience with when we're talking about mucosal immunology is, you have these specific receptors and markers that help cells traffic, but it does depend on the infection and, of course, the organ. But the cells do traffic from site to site. So in this paper, I just don't think that, while they do show an increase using, it's a good idea, but they don't, in my opinion, give enough, and they need to combine chemokines, and they could take advantage of some other time points to really look at, what other things they don't really measure right after the challenge. Why not draw blood and look at the cells that are trafficking at that point? Right. So let me see here if I've gotten through. Where are we at now? We can talk more about.
I think you're okay, actually.
I think I'm okay. Yeah.
So this is your shtick, Stephanie, right? Mucosal immunity. This is what you work on, and this is what you want to do for the rest of your life, right?
I would love to. This is what the area of mucosal immunology, but in the context of viral infection, I think we don't know enough about how viruses induce the immune response specific to mucosal immunology. And another thing that is a whole nother thing we could talk about, but we know that estrogen and hormones impact how mucosal immune system responds. I think they do show that HIV infection is actually increased in women who take estrogen-based birth controls. So another thing they don't control for is, what part of the cycle are these monkeys at, because that is going to definitely contribute. And another thing I'm interested in too.
Cool. Well, you have to come back and tell us about that sometime.
Yes.
Let us go right to picks since we're...
I just want to I'll make 1 minute's worth of comments about
Yeah.
readability.
Oh, yeah, sure.
I
Of
think
course.
I've talked too long.
This
most
paper,
likely.
That's okay.
evidently this paper is not copy edited, or this journal isn't copy edited. There were a lot of mistakes like that.
Yep.
P-values in the abstract was very misplaced. I thought they could have done a much better job with the figures. For instance, just in the very first figure, the 3 ones across, they don't have the same scale, and they could use a scale of 6,000, and we could see these graphs
Right.
much better. And the second part of that figure, there's all these pie graphs and arcs and stuff, and they don't describe it adequately in the figure legend. Another figure, the one that's the really nice one about the protection, it takes up half the page, but the graphs are really small. If they had done their 5 graphs vertically as a 1 column, they would all be bigger, and we would see a whole lot more. And then finally, just one example of the copy editing. They say, we see strong protection against challenge with the use of a DNA-only immunization regimen. And
So they
Alan had a
deployed the
comment.
troops to do the vaccination.
Right.
Regiment,
So
yeah.
there you have it.
All right. Very good. And let's do some picks of the week. Alan, what do you have?
Oh, let's see. What do I have? Let me scroll down past all these emails we're not getting to. Oh, yes. So Hook's Micrographia, which is you've probably all heard of this or encountered it at some point. It's a really seminal book of microscopy. I think it was published in 1655 or thereabouts. And Hook, of course, was a pioneering microscopist. He produced this book, which was, I believe it was the first best-selling science book ever. He took his microscope, one of the first microscopes, and just looked at a bunch of stuff and described it. And it's the artwork is just amazing, and it's always been just this really neat thing to look at. And now there's this nicely organized digital collection of all of the plates from Micrographia, courtesy of the National Library of Wales. And it's you can go and look these up, and they're in the public domain. So if you need to illustrate something and you need, for example, a micrograph of a louse, very nicely drawn by Robert Hooke, you can look that up and download a fairly good sized picture.
So he himself did the illustrations?
I believe he did.
God,
I love the titles.
you're amazing.
Of Gravel and Urine,
Yes.
of the Wandering Mite. This is so cool. Nice. It's beautiful.
Yeah, they are beautiful.
All right. Thank you, Alan.
Sure.
Rich Condit, what do you have?
I just finished reading a new book by David McCullough called The Wright Brothers. Alan, you would love this.
Yeah, I have to do this.
Oh, it is awesome. David, well, I've picked a couple of David McCullough, or at least one David McCullough book in the past. That was The Great Bridge, about the building of the Brooklyn Bridge. And he's just an amazing author. All of his research is from the primary literature. You know, this is all newspapers and correspondence among the Wright brothers. And wonderful photographs that he's found. And he just tells the story in a delightful fashion. And these guys, neither of them ever went to college or anything. There's a couple of interested guys, but they're scientists. And they studied how birds flew, and they designed their planes to reflect that, and they studied how to fly. You know, it's not enough just to build a machine. You got to actually learn how to fly it. And they practiced, and they did experiment after experiment after experiment, and failed and failed and failed, and they got it to work. And it's just an amazing story. It's great. And I just discovered today that there's, ordinarily I don't care about audiobooks, all right? But there's an audiobook of this, and it's actually narrated by David McCullough. And you recognize the voice because he narrated the Civil War.
Yeah.
All right, so that's good too.
Nice.
So I really highly recommend this. Great read.
Hey, Rich, did you see The Martian yet?
It comes out October 2nd, I think.
It's not out yet?
Unless it's, I don't think so.
Right.
My recollection
I
is
think
it's October 2nd.
some people have must be gotten bootleg copies because I heard it on a podcast, people talking about it. Oh, well. Okay, I'll ask you in October.
Yeah.
Kathy, what do you have for us?
I picked an article that's open access from your august institution, Vincent, from Columbia, by Oscar Sahin, or from his lab, and a video that was in the New York Times, which kind of autoplays, so I can't go back and check it at the moment.
Right.
But it's all about how spores, and when they take up water, can give you energy. So these people have built a little thing that raises and lowers some shutters, also something that drives wheels on a small car, and all from bacterial spores taking up water and the function of evaporation playing into that. So the video from the New York Times is really cool, and then, as I said, the paper's open access, and you can check out all the figures.
Cool.
And
This is neat.
Yeah.
And the video is very cool. I watched it earlier.
Yeah. Stephanie, what do you have?
Yeah, so I have a article by Carl Zimmer in the New York Times. I think it came out yesterday. And the title is That Stinky Cheese is a Result of Evolutionary Overdrive. And they discuss the molds that are used to make cheese. And they talk about how basically molds, that there's horizontal gene transfer, and some of these have up to 5% of their total genomes are DNA from other species. So it's really just talking about how these molds have evolved to make different cheeses, and it's just a really it's food and science combined, which I enjoy.
Do you like food?
So
Like
cheese is cheese is a gain of function experiment.
Yeah.
Well, these cheese, yes. So
Yes.
apparently we shouldn't be eating cheese. It's gonna.
No,
You know what, Rich? It's everywhere.
I know.
Gain of function is everywhere. So
I
the
know.
notion of what we doing being an issue is crazy.
Well, looking at this big thing of Roquefort's making me hungry.
It is.
Yes.
Roquefort is great.
But then if you scroll down a little more, you may not be hungry once you see that mixture of cheese-making fungi.
That's okay.
I don't
They're
know.
pretty.
I might just eat that.
They're
There you
pretty.
go.
I've always wondered about this stuff.
Do you know this program, How It Works, right? Have you ever heard of that? How things work?
Oh,
yes.
yes,
Oh.
yes.
I forgot, but I saw one once which they talked about how you make these cheeses, and they did a Roquefort. It was amazing because they have to make tons of these wheels. They have to make the mixture, and they pour it in, and then it has to ferment, and they have to take it out and then cut. Oh man, it is amazing.
So do they actually seed it with some of this mold?
Yeah, they seed it. It's very cool. It is just awesome. All right, that leaves me, and I have a pick that I mentioned last week. So Ben Fensterheim is an MD PhD student at Vanderbilt who I met when I visited not too long ago, and he does interviews with faculty on the Vanderbilt Student Radio, which is WRVU. And when I was there, he told me this, and I think we had lunch, and I said, "Why don't you interview me?" Yeah, nice guy, huh? And he said, "Sure, let's go." He said, "It's free," and we went to the studio and we sat down. It was a lot of fun. That's where I discovered Dengue Fever, the group. So I got him to post our interview, so it's on his SoundCloud site, and he has a couple of other interviews there with faculty. He interviews faculty of various sorts, and I thought it was cool because he fades in and fades out. He does advertising, and it was a lot of fun sitting there in the radio station with big mics. I mean, I guess I do that with podcasts all the time, but for some reason, being in a radio station, it's not even a real do they still have a transmitter? I think he said they sold the transmitter, for $5 million, and now it's just an internet radio station, but they still call it WRVU, which is funny. Anyway, it's a little self-serving, but you should check that out. And we have a listener pick from Reed, who writes, "Dear TWiV team, I'd like to suggest a new podcast called The Benchwarmers. As a listener pick, the podcast is hosted by graduate students and centers around life in the sciences from the perspective of graduate students. The second episode, OSH*T, features interviews asking current and former students about the most expensive thing they've broken in the lab and features some excellent stories. Does anyone on the TWiV team have a good story along the same vein? Thanks for the company while I'm plating cells in the hood." Well, I'm sure we all do, but we'll have to save it for another time because we've gone long enough now. Okay? Everybody could come up with stories, right? Maybe we could do an episode on lab stories.
Yeah.
Sure.
Yeah.
Lab stories, yeah.
You can find TWiV at iTunes and also at twiv.tv. And, of course, if you have questions or comments, please send them to us, twiv@twiv.tv. Our guest today has been Stephanie Neal. She's at Ohio State University. You could also find her at AgSci Today, agscitoday.com. And that podcast has a Twitter account, AgSci Today, and also a Facebook page, facebook.com/AgSci Today. Anything else I missed that you want us to mention, Stephanie?
No, I'm just really, really appreciative to come on here. I tell people this is like what probably people feel like when they go on a TV show or something. I've been listening to you guys for so long, so it's just been really, really great, and thank you so much.
You're welcome. This is better than TV.
Oh, but of course, it's science.
Dickson Despommier is at verticalfarm.com, and he's left us, but we thank him. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure enough, always a good time.
Does this keep you brain stimulated doing this?
Oh yeah. Oh yeah.
Keeps you in virology, right?
Yes, it does. Well, I'm a newbie to this retirement thing, so I don't know. This is I think going to be one of my main ways of staying into it. But I also have to, I got some editorial duties that'll help as well, and I want to visit the university now and then, and go to some research conferences and that kind of stuff. But it's all good.
You could go to ASV. We can do a TWiV there.
Well, that's what we're supposed to do, right?
You can be that cranky old guy in the back who asks the annoying questions.
Right. That's right. Alan Dove can be found at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.